Thanks, Ed, for inviting me to talk about low-dose dose response for the antioxidant response element activation. So it's been touched on a few times before, the critical cytoprotective pathway. Just a little, such a critically important pathway. Reactive oxygen species, ROS, are generated during respiration. They're quenched by a host of controlling enzymes that deactivate ROS. There we go. So let's see. So also important is the fact that ROS are involved in signaling pathways regulating cell survival. Pathways activated by ROS involve the alteration of protein kinases by the kinases which phosphorylate critical amino acids that change enzymatic behavior in many enzymes. So they can be involved in changing cell fate, cell behavior through this mechanism. That's part of the process of signal transduction using small modulations in ROS levels in redox state in cells. So they can change the behavior of cells from proliferative to static and back again. But at much larger levels, ROS, of course, can trigger apoptosis when levels of redox dysregulation become substantially elevated and start to cause cell damage, and can also, that apoptotic pathway can shift toward necrosis at even higher doses, which involves inflammation. So in this critical capacity of cells, all eukaryotic cells, to regulate redox levels, the antioxidant response pathway controlled by the NRF2 protein is critical. So under basal conditions, just as a review, the NRF2 is sequestered in the cytoplasm according to the current predominant model. The NRF2 is bound by KEAP1 dimers, which have a number of critical cytosine, cysteine residues that can react with oxidative chemicals and reactive oxygen species. The breaking of disulfide bonds can cause conformational change in proteins, including KEAP1. That can then, oops, sorry about that, that can cause a release of NRF2 from the KEAP1 dimer, which under normal conditions, when the dimer is present, it facilitates proteasomal degradation. Very rapid process of generating NRF2, and you're destroying it rapidly. But in the presence of sufficient ROS oxidation, we release the NRF2, and then it frees it to enter the nucleus, trigger the antioxidant response element, inducing gene expression of 250 or more cytoprotective genes. Importantly, drugs and natural molecules can activate the NRF2 pathway, allowing it to release and trigger ARE activation. And here I'm showing you an important triterpene anti-inflammatory agent, CDDOIM, mentioned previously. This is a very interesting chemical. It's one of the most potently anti-inflammatory agents known. So, just to highlight why I was interested in the dose-response relationship for ARE activation. is because it can have a profound influence on many cellular processes and pathologies. And the one that I focus on a lot is carcinogenesis, and I'm intrigued with whether or not the multi-stage somatic mutation, or MSM, model is actually correct. And this is an interesting experiment that was reported about 3 years ago by a group at Dartmouth and Johns Hopkins. Really a culmination of about 20 years of research on aflatoxin chemoprevention. So we're looking at rats who are given a 4 weeks dose of aflatoxin, a very highly potent mutagenic carcinogen. Enough aflatoxin to cause 100% of them, or very nearly 100%, to have liver tumors. So we're going to compare that with rats treated the same way, early on, 4 weeks of aflatoxin exposure, but also CDDOIM, very low doses during the period of aflatoxin exposure, and we follow up and measure DNA adducts, including the highly mutagenically potent form of DNA adduct that's 10 times more potent than the N7 guanine, liver foci, and liver tumors. We look at DNA adducts. The interesting thing here is, yeah, DNA adducts are reduced, but they're not totally reduced. They're not eliminated. About 40%, including of the most potently mutagenic adducts, remain. When CDDOM, the anti-inflammatory agent, is co-administered compared to just aflatoxin alone. So if we look at foci induction, we see that in the aflatoxin exposed rats, we get a dose-related increase in premalignant foci. But when you add the anti-inflammatory agent, you nearly ablate the foci generation, which is typically considered a mutagenically induced preneoplastic state. This is quite remarkable. Moreover, the reduction is highly statistically significantly at odds with the somatic mutation multistage theory under the linear no threshold assumption regarding increased risk of mutations, and likewise for liver tumor induction. Remember, aflatoxin exposed mice, nearly 100% of them get liver tumors at the dose used. But if you add the anti-inflammatory agent, the liver tumors were completely ablated. So this is a paradigm shifting experiment. This is a direct challenge to the somatic multistage theory because the results are highly significantly at odds. So this is the kind, and we're doing this with an NR2 ARE activator. So that's telling you, wow, this plays an important role in lots of important biological processes and pathology. So that's what intrigued me about the low-dose dose response for activation. What caught my eye back in 2012 was a paper that reported the results of a huge number of chemicals screened in a high throughput sense for ARE activation as well as other endpoints in a collaborative effort by NCI and NIEHS. And in this case, they were looking at ARE activation by hepatotoxic chemicals, about 3% of which were shown to be active in an assay that involves either of two types of reporter for ARE activation. We're using human HepG2 liver cells. And what I was interested in was some patterns in the plotted data for 15 of the chemicals. That looked intriguing because it seemed to be, so intriguing that I approached the authors to see if I could reanalyze their plotted data, their raw data, and this is the result. I was able to show that for 9 chemicals that were clearly positive in the assay, they reported some marginally positive chemicals as well. We're talking about, there were so many replications done at each of 12 different concentrations. So we have a very rich data set, over 500 data points. And what was apparent is a J-shaped dose response for activation of the ARE receptor. All 9 chemicals show a statistically homogeneous negative initial approximately linear slope, and then the chemical responses become heterogeneous when the response becomes net positive. So you're seeing an average of those responses here. But in this portion, they are statistically homogeneous, and notice that includes the inflection point of around 1 micromolar. Highly significant negative slope. So here we have a J-shaped dose response for this critically important ultrasensitive molecular switch. These are the analysis of covariance results showing the homogeneity of the net negative activation levels. And we get heterogeneity at the more positive responses. This also was the case when I analyzed the second of 2 reporters used using somewhat less positivity in terms of response. Only 5 chemicals were clearly positive, but nevertheless, highly significant initial negative slope that's statistically homogeneous. So, in terms of mathematical modeling of the response of ultrasensitive molecular switches, a J-shaped type of dose response was predicted as being possible, and the evolutionary mechanism, rationale, would be that for such an critically important ultrasensitive molecular switch, you want to avoid triggering that switch in marginal situations. There's a cost to triggering the switch. And so this may be a typical pattern for other ultrasensitive molecular switches. And I'll just show you an example of another one that I modeled recently in a paper in Risk Analysis and Press. Previously reported data where the authors didn't actually model the response. But here we're talking about activation of the heat shock protein response. Looking at heat shock protein 70, and you see the black dots are the induction as indicated by a reporter, very similar to the ARE type of reporter, at least separase reporter. An apparently nonlinear response that's ablated at much higher energy levels of the laser that was used to induce this in the context that has to do with ophthalmology and treatment of eyes and determining what laser levels are not cytotoxic, what amounts of energy deposition are safe. So it drops off at very, very large temperatures. They only report the data starting at about 42 degrees centigrade. But the cytotoxicity has a mirror image, very threshold-like with an abrupt and apparently kind of a linear decrease in cell viability. But again, the data were only reported graphically down to about 41 degrees. But in the text, they report that at 37 degrees, the viability was lower. And the difference is highly statistically significant. So in effect, what you have is an inverted J-shaped dose response for viability as a function of laser energy deposition and associated increased thermal energy. So that's another example of an ultra-sensitive molecular switch with highly important roles in cytoprotection. So the conclusion is that the ARE response is J-shaped, and that can have interesting implications. And one that fascinated me that I'll just mention is the potential implications for low-dose dose response for chemically induced tumor genesis. It becomes relevant if instead of the somatic mutation theory, it may be the case that NRF2 ARE activation, when it's sustained, chronic, and involved and associated with cytotoxicity and inflammation, may be sufficient to drive tumorigenesis. So that's a hypothesis. In that case, then, given the J-shaped dose response for the activation process, it would follow that oxidative chemical exposures, including for many genotoxic carcinogens, would also have a J-shaped dose response, or a highly nonlinear low-dose dose response. So, of course, this is in contrast to the multi-stage somatic mutation theory, which predicts a linear dose response. And the aflatoxin data, the experimental results that I showed you, are a hint that something seems to be wrong with the MSM theory. In a profound way, there is and has been over the previous decades an accumulation of evidence pointing to some problems with that theory. Most recently, in addition to the aflatoxin experiments, was a study published in Science 4.5 years ago, looking at deep genome sequencing of normal human eyelid biopsy. So this is deep sequencing, meaning cell-by-cell analysis of mutated oncogenes. And the conclusion was that there are so many mutated oncogenes in the normal cell that it questioned as to whether they play a role in causing tumors. The hypothesis that I've put out there supporting the possibility that NRF2 AR reactivation might be able to drive tumorigenesis depends on an alternative model of tumorigenesis, which is driven by epigenetic change, but also can involve mutation. And but the basic gist of the hypothesis is that if epigenetic changes, in this case microRNA expression profile, can serve as a basis for driving typically observed increased rates of tumorigenesis in chemical exposure experiments, then you have this interesting implication in terms of dose response. MicroRNA, of course, is created in the nucleus and essentially edited to 22 base pair, roughly, stretches that get loaded into a position in the cytoplasm where they can interfere with the expression of protein from messenger RNA. Translation. And the importance there is that it happens very quickly. These are little pieces, little molecules. They're much faster than DNA transcription feedback mechanism. And that's why there has been an increasing recognition over the last 10 or 15 years that microRNA expression profiles play profound roles in embryogenesis and also in adult tissue phenotype and pathology, including regulation of the cell cycle and including modulating activities of oncogenes. It's just that the microRNA act as barcodes in a distributed system that orchestrates genome expression in a very fast way, since the host of many genes can include many different species of microRNA, essentially barcoding the messenger RNA that gets produced. The microRNA are homologous to that, so they gum up the translation into protein. They can also cause the degradation of messenger RNA. So it's a very quick acting response that changes cell behavior. So how can this, also dysregulation in microRNA expression profiles have been tied extensively to and diagnostically to different kinds of cancer. That's been an exploding area of research and now development of therapeutic approaches based on microRNA. So this is the theory that added the NRF2 ARV activation component to a previous theory that was microRNA driven concerning tumor genesis that I published in 2013. The basic concept is that normal cells in different tissues can undergo adaptive hyperplasia under different circumstances. This is a normal process. Cells are recruited into that process, and then that process terminates when it's no longer needed. And the idea is that adaptive hyperplasia comes in two different flavors. One, regenerative repair, involving requirements for migration of stem cells to points of damage. And the other one, a protective form of adaptive hyperplasia, which involves tissue thickening in response to a toxic gradient or mechanical stress or increased metabolic demand. So one acts fast, rapid, and one is slow. And the idea is if you break the termination signal transduction mechanism, even by mutation, then the cells just keep going. So it's a form of defective wound repair. It's the old name for this kind of a theory, but this is a proposed molecular basis for it. And if you're involved in the rapid flavor of adaptive hyperplasia, the cells keep going until they run into their Hayflick limit, and C2 becomes genomically unstable, and you get malignant tumors. Whereas the slow version corresponds to benign tumors in each tissue. So it's a different set of microRNA for each tissue, and that's the underlying theory. The way that this differs profoundly from the multi-stage somatic mutation theory is that that theory posits a generally stable fraction of all cells as being the target stem cells involved in the process, except for a few hormonally controlled tissues. So you have a certain stem population size that's a little bit age dependent early in life, but for adults, it remains fairly constant. This theory differs tremendously because it says that only during episodes where you recruit cells into adaptive hyperplasia do you actually increase the population size of that subset of stem cells that are critically involved that can be subject to destruction of the termination transduction. And so you see this episodic population of cells, and that explains the background rates of tumor incidence for different tissues. NRF2 activation plays a role, I'm hypothesizing, insofar as that if there's evidence indicating that NRF2, as has been mentioned, ARV activation plays a role in changing the cell's metabolism to the Warburg form of metabolism that's required for cell proliferation. And that metabolic scheme, most of the energy is shunted to generating the precursors to the macromolecules required for cell growth or cell proliferation, rather than to energy production, ATP production. That's called the Warburg and metabolic state. And NRF2 and ARE activation plays a key role in making that transition. So the basic idea is that you either have physiological or cytotoxic stress. You get non-inflammatory stem cell recruitment or inflammatory, and they lead to either tissue repair or tissue expansion normally. With the sustained Warburg metabolism being associated with the malignant pathway, but intermittent for the protective adaptive hyperplastic pathway. So posited in the new model is that chronic ARE activation can be associated with metabolic stress or cytotoxic stress, but then it resolves. And so you get this burst of basically increasing your effective stem cell population, and that's enough to explain dose response for increased tumor incidence in a bioacid type situation. That's basically the idea, and it's just one way, probably many, that a J-shaped dose response for antioxidant pathway activation may have important implications for most of the compound. Thank you.
Thank you.
Interesting idea. On the key part for the NRF2, I know that there are cysteines or cysteines that are around here. Do they have to get oxidized before that release occurs?
Evidently, there are. It's been mapped, and it's shown that only certain of the cysteines within the KEAP molecule, which has a higher level of cysteines than most other proteins, play a role in activating the antioxidant response. So, yeah, what you're doing is the reactive molecules are breaking a cysteine-cysteine disulfide bond, changes the protein conformation, allows the NRF2 to escape.
So if you've got a redox change in the state, you could probably get more oxidative state, then you would oxidize the cysteines. That would release the NRF, and that makes the sense, right?
That's how the
so the signaling, that was your point about the early ROS signaling, especially we see a lot of that in the hormetic kind of phenomenon, right?
Yes, so the antioxidant response evidently has an overall nonlinear pattern to it, which evidently begins with a J-shaped activation response. But cells either undergo small or moderate changes in ROS activity as a way to transduce phenotype change signals. So that's a natural thing that's ongoing all the time. But then more and more levels of oxidative stress trigger a large amount of ARE induction, and then the suppression enzymes are generated. But then even greater amounts switch the system. They flip it toward assisting apoptosis and flipping from apoptosis to necrosis.
Right. And then one other thing, you mentioned the Warburg metabolism.
Yes.
So based on that theory, the mitochondria are usually defective or defunct. You know, they're usually damaged, and that's when you shift into the glycolytic pathway. The Warburg metabolism, is that what you're talking about?
No, my understanding is that it doesn't involve any defective mitochondria. It's simply a shift in the metabolic program. It's a shift toward a revised program that shunts most of the energy, the ATP production capacity, into generating macromolecule precursors, nucleotides, amino acids. It shunts that energy pathway toward creating those molecules, taking most of that ATP generating energy to produce larger molecules, simply to sustain cell proliferation. This does not involve a defect. It's something hardwired.
All right. Well, my understanding, well, some of my reading was indicated that there's mitochondrial damage, which causes an excess of ROS, and then the ROS could stimulate the NRF2 pathway, which fits into your theory here, and that that damage to the mitochondria, and I'm not sure what the switch or the signal is, but causes the switch to glycolytic pathway, the Warburg pathway, simply for cell survival. The cell needs energy, and if it can't get it through the Krebs cycle, then it moves over to the glycolytic pathway. And so the two concepts sort of fit together.
Yeah, except that I'm not so sure that the cell proliferative supporting shift involves mitochondrial pathology. There is a change in mitochondria number associated with the Warburg shift. But this is a normal cell condition that's encountered whenever tissues have to engage in adaptive hyperplasia and have to generate material for cell growth.
I have a quick question. You mentioned NRF2, RS, and apoptosis, even necroptosis. So how do you think NRF2 and ferroptosis?
In what was that second word? Which kind of
Therapy.
therapy?
Yeah. Yeah.
Of a therapeutic application?
Yeah. Another.
There's a different flavor of apoptosis?
Which one about the
I see. It's a different flavor of it. So there are some different flavors of apoptosis. That's one of them that I know. There's a couple others. And that's different than necrosis, which releases the inflammasomes and cytokines that trigger inflammation and recruit immune cells. Because necrosis is naturally what happens in microbial infection, where a lot of this evolved to be prepped to engage in tissue repair and tissue maintenance in the event of a microbial infection. You've got to regenerate a lot of tissue quickly, faster than the microbe works. So I'm not familiar with literature that distinguishes ARE activation component of different apoptotic pathways. It's just centrally involved in first suppressing apoptosis in general. That's what happens when you trigger the ARE response. Basically, you're prepping the cell to survive a cytotoxic redox dysregulation. So to anticipate the kinds of damage that occur and prevent unnecessary loss of cells on a hair-triggered kind of a basis. So you're buffering the typical ROS effects that could occur at higher levels of dysregulation. And there's a point, a tipping point, where all of a sudden NRF2 ARE activation flips and facilitates apoptosis. So it's involved in both legs of that. So, but its initial response, interestingly, is this J-shaped component of triggering the ARE activation. So there's some resistance to it, and that may have some interesting implications in terms of physiology, pathology as well.
Thank you.